Make a start. So my name is Virginie, and I'm the Research Infrastructure Project Manager at EURORDIS, and I also chair the Patient Advisory Council of the RD Connect project, and I will be moderating the webinar today. So we will have two presentations, each lasting about 20 minutes, followed by a question and answer session. But we will take your question after each of the presentation, if that's okay. It's just to ensure smooth running of the webinar. But in the meantime, if you think of the questions, and you can always type it in the chat box that's on the left inside of your screen. So the first presentation will be given by Libby, who's the project manager of RD Connect, and after Libby, Sergi will be talking about the genome-phenome analysis platform. But before we start, we wanted to show you a little video that's very well done and explain a little bit what RD Connect is about. So maybe we can start the video.
Rare diseases can severely affect quality of life and life expectancy. 1 in 17 people suffers from a rare disease. This means in Europe alone, an estimated 30 million people are affected, such as Sophia, who suffers from congenital myasthenic syndrome. Most rare diseases have a genetic origin. Thanks to recent progress in genetic sequencing and innovative technologies, researchers can now study rare diseases, improve their diagnosis, and develop more effective treatments. Because of their rarity, research and therapy development for rare diseases can be challenging and requires that researchers join efforts and exchange data across countries. RD Connect makes that possible. RD Connect is an online platform that allows researchers all over the world to share and exchange disease information and genomics data with other researchers in a safe and secure environment. Using the bioinformatic tools available in the RD Connect platform, doctors looked through Sophia's genome and pinpointed the precise defect causing her disease. They saw that she had inherited one defective gene from each of her parents. This helped doctors to get Sophia the right treatment and to explain to her parents the risk of having another child with the same condition. RD Connect also provides researchers with secure access to databases containing clinical information about rare disease patients, as well as patient registries, biobanks, and biosamples such as blood and DNA. To ensure protection of patient privacy without compromising research and drug discovery, our team of experts on ethics, legal and social issues provides guidance to researchers and works on the most appropriate models for data sharing. Rare disease patients and families are actively engaged in RD Connect to ensure that their needs, concerns and expectations are addressed. For more information, visit rd-connect.eu.
Thank you. That's great. It was a good video, and as you can see, Sergi added a link where you can find the videos in many different other languages as well, so that could be helpful. So now we can start with the first presentation. You can see it on your screen. So Libby, thank you. If you want to make a start.
Yeah. So thank you, Virginie, for organising this webinar to start with. RD Connect is a project that's been going since 2012, and so it's funded by the European Commission. And hopefully that video has given sort of a brief overview of everything that RD Connect does. And I'm going to hopefully try and go into a bit more detail about some of those things, and then Sergi will concentrate on specifically the genome-phenome analysis platform that you saw mentioned there as well. So just as a bit of a background, I'm based in Newcastle University, where the coordination office for RD Connect is, as it's coordinated by Professor Hans Lockmuller. So at the heart of RD Connect is really the idea to sort of facilitate research in rare diseases. And For many reasons, research in rare diseases can be more challenging than in common diseases, and that really comes down to the rarity of the patients themselves, and that means that there's less data for research, there's less samples provided, it can mean there's less interest from pharma and academic researchers, it's harder to get a diagnosis, it's harder to confirm a diagnosis, and it can be harder to have enough patients to conduct trials and have better treatments. And many of these problems don't just apply to one area of rare disease research, but really can come down to many different areas and can be across all diseases. And a lot of them really come down to data itself, and that there is a scarcity of data and also a lack of options to reuse this data. And so that can be for lots of different reasons. There's particularly, within Europe and then within the EU and outside the EU, there's lots of different regulations that stop data being shared. Privacy protection issues, there's sometimes just the lack of infrastructure for being able to share that data, lack of standards and interoperability. There's also still some reluctance in certain areas of the academic community to share data before they've had it published. And there's challenges of linking data that's stored in different areas and different types of data. But what RD Connect is trying to do is really promote the benefits of sharing and give some solutions to some of those problems. And in essence, the sharing of data helps overcome the rare disease problem. It helps make it easier to have larger cohorts, easier to power trials, easier to diagnose patients. And also it has other sort of benefits, like you can reduce the cost if you share data, the cost of research, and you can reduce duplication of effort, and it can be easier to validate your results. And sharing also just enables that sharing that communication between experts, the patient community, different experts from different areas. And so when we're talking about data and the sharing of data, what do we really mean? And it is broad, and within RD Connect there's lots of different types of data, but primarily we're talking about genomic data, so that's the data that comes from the sequencing of DNA, and phenotypic data, and that's the clinical description of the symptoms that people experience, as well as data that's obtained through natural history studies and clinical trials, and also samples and the data linked to samples, so that can be the data sort of that you give with your DNA samples or tissue samples, biopsies, that kind of thing. And so kind of traditionally what happens to the data of a rare disease patient. So a single patient could provide their data to many different projects at once. So you could go to clinic and provide a sample into a biobank that is stored in that biobank with some data about the patient. At the same time, you could sign up to a registry. That data is stored somewhere else. Then you might take part in a clinical trial. That data is kept by a pharmaceutical company, for example. And all of this data is housed in different silos, in different sort of contained areas, and it can't talk to each other, and it can't be shared. And so what RD Connect is trying to do is change that in one way or another, and that is through developing an integrated platform for connecting databases, registries, biobanks, and clinical bioinformatics. So that's quite a sort of wordy title, and hopefully we can break it down a little bit. And so back in 2012, when the project started, EURORDIS, the International Rare Disease Research Consortium, also was being founded, and they had two main objectives. That was to deliver 200 new therapies for rare diseases and the means to diagnose most rare diseases by 2020. Now these objectives have actually recently changed, so they've met them. Well, they've met the 200 new therapies for rare diseases already, but RD Connect is still committed to working with EURORDIS to help reach these goals. I don't have the exact new goals in mind. I realised that I didn't have the new ones, but they're focused more on decreasing the time to diagnosis as well as increasing the access to therapies. And so this is quite a busy slide, but hopefully it sums up RD Connect all at once. And what I wanted to draw attention to the most is the sort of really the three statements at the bottom. So there's three main flagship areas to RD Connect, and that is the genomics analysis platform, or the genome-phenome analysis platform, as we're calling it, and that's really what Sergi is going to focus on after me, so I won't go into too much detail about that. And there's also a sample catalogue that helps make samples more findable, and a directory of registries and biobanks that helps increase the access to that data as well. So this is just a quick screenshot of the analysis platform. As I mentioned, this is really what Sergi is going to discuss, but just to show that there's a lot of information in there that can be accessed by researchers very easily. And I wanted to just quickly go through the workflow of this platform and sort of really focus on the the bottlenecks within this process. So the genomics analysis platform is taking data from the rare disease patient and bringing that through lots of different areas, whether that's through biobanks, through sequencing data, and then bringing it all together at the other end. And there's lots of sort of points where you need to think about standardization and infrastructure to make this possible. And so one of the first issues or areas that you really need to think about is the consent when that sample is taken, or when that patient agrees to submit their data to something, to a registry or to another research study. And it's important that those consents allow for data sharing, allow for data storage, and allow for that to be international. And that's one of the things that RD Connect's been looking into, along with many other groups. And then there's other standards that need to be thought of in terms of how you collect that data. So in terms of phenotypic data, how you describe something. So you know, one doctor might describe weakness in the muscle, another doctor might say muscle weakness. That sort of seems quite straightforward to us that it means the same thing, but if you're looking at large data sets, it can be hard to combine those things. And similarly, when you're looking to genomics data, it's important that that's described in the same way, that it's analyzed in the same way, and that's something that we've been working on too. And then on top of those standardizations, you also need the infrastructure for these things, for the sharing of samples and data analysis. And these are all things that we've been building in to the platform that we're building. So Sergi will go into more detail on sort of that platform and how it works. So I'm going to focus a little bit on some of the other areas, and one of those is in biosample sharing. So there's lots of biobanks in rare diseases, and the samples that are taken from rare disease patients are particularly valuable, but it's important that these are used. And so one of the things that we've done is create a catalogue for the registration of rare disease biobanks. So this has been previously referred to as the ID cards. We're moving towards calling it the Registry and Biobank Finder, so apologies that might be confusing just in these slides if both terminologies are used. But that means that biobanks are at least visible, and so we're working closely with the EUROBiobank network, which is led by Lucia at Monaco in Italy, and this is so all of those biobanks are now part of the RD Connect catalogue. And on top of that, we're working on sharing sample level data. And so this is more than just saying this biobank has, 200 patients with this disease, 200 samples, with this disease, but saying really that this sample has this from a patient with this phenotype, and that data might also be in the platform, and really linking things at that level. And that's what we're working towards at the moment. And so with all of these things, it's important to have that phenotypic data described properly in a way that's standard and that can be shared easily. Phenotypic data is collected in lots of different ways. It is in our genome-phenome platform. It's also collected in patient registries. It's collected alongside biosamples. And so we're working to make sure that all of this data is collected using the FAIR data principles, and so that it's findable, accessible, interoperable, and reusable. I'm just going to walk you through a brief example of why we need this and sort of how it can benefit research. And this is an example from Marco Roos from Leiden, who's really pushing this work forward. So this slide just shows how you can be collecting the same things, but they're not described in the same way. So I'm just going to highlight the age and date of birth here. So one place collects the age at baseline in years, somebody else collects the exact age at visit. Here it doesn't even say whether that's in months or in days or in years. And then you could have somebody else collecting the date of birth itself. And while sort of on paper it sounds like this is all the same information being collected, if you just dump it all into a spreadsheet, you can't pull it all out at the same time. So what the FAIR principles do is try and make this computer readable, so that kind of data isn't linkable at the source. So this is a quick back of the envelope calculation that Marco's done to show how much time it takes to make that data integrated, interoperable, and then sort of how that can be reduced. So he's estimated that each data set, it would take about 6 months to make reusable, and you do that at least 5 times, and that would be 30 months per data set. And if we say there's 6,000 rare diseases, that would be 180,000 months for every rare disease, which sounds like a crazy amount of time. When with data is made linkable at source, it's not an instant process and it's not an easy process, so there is still some work to be done. but that work just needs to be done once. So you would just need to do that once to make your data set fair. That Mark was estimated would take about 6 months, and then that would mean that for every rare disease, this would just be 66,000 months, which still sounds like a lot, but there is an efficiency gain of 66%. Therefore, researchers can do more research and analyze that data faster. So the FAIR principles are just one way to try and to make data more accessible, and that's linked to the use of ontologies, which is another thing that we're promoting in RD Connect, and it's a way to describe phenotypes in a way that computers can read, and in a way that means they're standardized. So within rare diseases, we mainly use two ontologies. That's the human phenotype ontology and the orphanet rare disease ontology. So HPO, the human phenotype ontology, sort of describes phenotypes, and the orphanet rare disease ontology names diseases. So this is an example of sort of one arm of a tree of an ontology which describes limb-girdle phenotype. And so what's important about ontologies is that you have this tree-like structure, so that even if everybody doesn't go into the same level of detail in their description, you can still tell that they're linked. So if one doctor was to describe a phenotype as abnormality of muscle physiology, and another one was to say it was proximal muscle weakness, you would still know that both of these patients had abnormality of muscle physiology, even though they both didn't have that level of detail. You can still go back up the tree, and that's one of the really kind of powerful things about the ontology, to make sure that data isn't lost by different sort of levels of detail. And so just carrying on with the kind of registry work that RD Connect is doing, the online registry and Biobank Finder, the previously called ID cards, I mentioned before, has this information for biobanks, but it also contains information about registries. And this is more than just sort of a list of rare disease registries, but it really goes into a lot of detail about the information that they collect and the standard of the registry. So it has a disease matrix that shows the different genotypes that might be within the registry, and then it also has an area to collect various documents included with that registry. So that might be sort of the SOPs, the procedures for data access, the consents that are involved. So before you contact that registry, you can already see that, you know, the consent includes data sharing, or that they're going to collect a certain type of data. And that can be useful both for patients wanting to sign up to registries to see, you know, what's involved, but also for researchers and potentially industry who are wanting to organize clinical trials. They can really get a lot of information from these ID cards. And so important to mention how much EURORDIS have been involved in RD Connect, and how much the patient representatives have also been involved in all of this work. So the Patient Advisory Council is a group of 16 representatives, all volunteers, led by EURORDIS. And so this includes representatives particularly from rare kidney disease and neuromuscular and neurodegenerative diseases. And that is because at the start of the project, we worked closely with Genomics and Neuromics, which were two EU-funded projects that were generating data that the platform could use. And so we specifically have patients from those areas, and these experts are really involved in directing what RD Connect does and making sure that what we're doing is important for patients and not just for the researchers involved. And this is just a slide to show you who all of those members are, all from all around Europe, all sorts of different diseases involved, and chaired by Virginie. And as part of the PAC, the Patient Advisory Council, there is a terms of reference to make sure that that the work that they do is important to RD Connect, and that the activities that RD Connect are doing is important to the patients. And what's really important is that this flow of communication goes both ways, and the patients involved in the PAC are involved in different work packages so that we can really get their input, and that they meet at our annual meeting every year so that we can really get the feedback. And so as part of, I think this slide might have moved, but along with the work we're doing with EURORDIS, there's also a lot of work around ethical, legal and social issues in in RD Connect and around data sharing. And this work is led by Mats Hansen at Uppsala, but certainly is done in close collaboration with EURORDIS and other colleagues. And they've developed this, or written this, international charter of principles for sharing biospecimens and data. So as you can imagine, that within the arena of data sharing, there's lots of questions about what should and shouldn't be allowed, and what information should and shouldn't be provided to patients when they're making this decision. And this is some guidelines that have been developed to help that. Okay, we've already been through these, so I'll just move on. Okay, and so we obviously want to engage with researchers and clinicians, but we also want to engage as much as possible with the patient community. And so it's important that the patient community discusses these issues when they are speaking to researchers and clinicians that they come into contact with, particularly if joining a research project and there's, consents aren't mentioning data sharing, to ask those questions, and to help contribute to research by providing samples and joining registries if patients wish, and to help communicate the results of projects like RD Connect and of other research, both using the RD Connect and EURORDIS dissemination channels. And so the bottom line, and this is my final slide, is just that data sharing does benefit research, and RD Connect is working towards making data sharing as easy as possible for within rare disease. It helps in gene identification, in having more samples available, and ultimately accelerating therapeutics. And so those are just a few of the people involved. Sergi's on there, who you're going to hear from in a few moments. And then just to say thank you to all of the different partners within RD Connect here who are raising their hands for Rare Disease Day earlier this year. I think that's all from me. So I think we'll take some questions now. Is that right?
Yes, thank you very much, Libby, for this comprehensive introduction and explanation on the project. So to ask a question, you have two possibilities. I can see that some of you have already started to type them in the chat box, and I'll be happy to ask them on your behalf. Or you can also use the mic that's at the top black bar of your screen, and you just need to unmute yourself if you want to ask a question. So it would be great also to hear your voice, so please do not be shy. But in the meantime, maybe I can just take the first question. So Nancy was asking how widespread is the use of the FAIR principles for data sharing?
So I think it's becoming more and more widespread. I think it's not a straightforward process. So I think it's hard if data sets already exist to become FAIR, but I think it's more becoming more and more popular that new data sets are sort of built FAIR, if that makes sense. And it's certainly something that is being pushed by, say, the European Commission, and therefore sort of new grants and new research that's been doing will really be encouraged to abide by these FAIR.
Thank you. Yeah. And yes, Kirsten, I apologize, I should have said that from the start, but the webinar is indeed being recorded, and we will also circulate the link to the recording within a few days, a week maximum, when it's available. So people who registered but could not attend, for example, or even you can disseminate the webinar to your contacts as well. Laia is making a comment on the Ordo ontology. That's very interesting, and that she's developing a holistic ontology about rare diseases and providing a link, so that's helpful. Maybe, I don't know if Libby or Sergi, did you want to comment on the Ordo coding?
What I can say is that for phenotyping, we are currently only using HPO. There is an international consortium for human phenotypes ontologies, which includes HPO and Orphanet, among others, which are or which have developed a minimum set of terms that are convertible between these different ontologies. And then the final comment I can make is that for recording the diagnosis, the disease, we use the Orphanet ontology for that.
Thank you. Yeah, that's helpful. Nancy also asked whether, and I think maybe you did touch upon that, Libby, how can patients at least encourage people to use the platform, but can they use directly the RD Connect platform?
So at the moment, patients themselves sort of aren't granted access to the data within the platform, but they can certainly sort of access the registries and things through the ID cards. And as I mentioned, they can definitely encourage other people to use it, researchers and clinicians. It is something that's sort of been discussed, been thrown around, but at the moment we haven't come up with a solution to that as to whether they can access the data.
Okay, great. But we'll make sure to update them on that. But yes, as you said, they can talk to their clinicians and maybe the researcher they might have link with. So that's also a good way to participate in the development and to increase data sharing as well. I don't know if... yeah?
Maybe to expand on this, just a couple comments. That even if in the video you see that the person using the platform types Sophia, there are no names in the system, so they are just codes. So one way, so there are some privacy issues of having someone access its own genome in the system, because you wouldn't know which genome it is, right? Unless it goes through a secondary partner who controls this relationship between the codes and the actual patients. Having said that, we, as Livy said, we are thinking about ways of making these available directly to the patients, if not through RD Connect, through another system, but these are things that are still on the way, and surely all these need, if it can be a funnel through an association like EURORDIS that can gather the needs and what are the specifications.
Thank you. No, that was an important clarification to make in terms of the name is not, yes, it's a code, so that's obviously very important to be able to share the data with third parties and partners. I don't know if anybody wants to use the microphone maybe to ask a question so we can hear your voice, or we can also keep going with Sergi's presentation, and we'll always have the opportunity to have a question and answer session after. Is that okay, Sergi? Shall we go ahead? Great. Okay.
Yes, sure. So I wanted to ask, do you see my pointer, right? Okay, excellent. So I will talk basically about two different points. The first one is I'm going to describe a little bit, an overview, not too much into detail, the RD Connect Genome Phenome Analysis platform, and then I will show you how we have used this platform in a project we have recently done in collaboration with BBMRI LPC, that's a European project, to sequence and analyze 900 exomes. So first of all, I wanted to say that I'm from the National Center of Genomic Analysis. That's a center sitting in Barcelona. We do have a lot of sequencing capacity, but also a lot of bioinformatics capacity. And in the project, in RD Connect, we are basically contributing from the bioinformatics part, although, for example, in some cases, we do submit data that we sequence to the system. Okay, so I will pick up on the slide that Livy showed before, in which she described the overview of the whole project. And as she said, the project has many legs or many arms, and the platform we're trying to make is an integrated platform in which the genomic data is also connected to the biobank and the registry. So we have a system that when someone submits a data set to the RD Connect, it can say on which biobank the sample sits or on which registry this patient is registered, for example. So we try to have some kind of integration of this information. So it's not only tools which are separated between them, but also we try to integrate them to some extent. Then focusing more on the genome phenome analysis platform, I will say that the workflow that we are following is that on the first thing that the clinician does is send a sample for sequencing. As I said before, it doesn't have to be the SENAC, it can be any sequencing center or lab in the world. And then that this data, the raw data, it's transferred to the European Genome Phenome Archive. That's a controlled access data repository that is used for storing raw data from human beings. We process this data through a standard analysis pipeline that I will show a bit afterwards, and then we integrate this information together with the clinical information, with the phenotypic information in the system, and then we provide analysis tools to the clinician researcher so that they can try to identify which are the causative variants from these patients. That's the main page from the platform, from the RD Connect platform. You can access that at platform.rdconnect.eu. You have links to some of these components that Libby showed before, like the biosample catalog, which I think is not yet active, but it will be soon, the ID card, so it's the finder of registries and biobanks, but also the genomic information and the phenotypic information, which is what I mentioned before. It's really tightly integrated in the system. So how do we collect the phenotypic information? We use a software called Phenotypes. We have our own instance of Phenotypes. It's a software developed by Mike Brudner's team in Canada, in Toronto. And we use HPO terms to codify for the phenotypes. And then we use OrphaNet, Rare Disease Ontology, and Amoeba identifiers to classify the disorders. What we do is set up very specific templates for groups of disorders so that it is easy for the clinicians to enter the phenotypes of a patient. And then also we can record the relationship, the families in that system, so we know that if we get 3 exomes, we know who is the father, who is the mother, who is the son, for example, and we know which one of them is affected, what is the inheritance mode, etc. So we have lots of information standardized with codes. A nice thing about doing it this way is that someone entering the information in English is compatible with someone entering the information in Spanish or Japanese, because HPO is already being translated to several languages. So we can display this information, these templates in different languages, so different doctors from different countries can make use of that information. Then on the side of raw data, I'm not going to go into details, of course, here, but I will just say that we do process the data through a standard analysis platform that goes through different steps. Most of it is using software that is available publicly and that has been thoroughly tested by many groups. Among us, last year we published a paper and we compared several different pipelines using different mappers and colors. And then we assessed how sensitive and how specific were our calls from RD Connect. As you can see here, we are very sensitive for SMBs and indels, so we are able to detect everything that we should be detecting, and we are also very specific, meaning that we don't detect too many things, so we don't give also too many false positives. I have to clarify those, that although we are very good in doing this, so we are applying, as I said, standard analysis systems, the analysis is not a solved issue. And we can do very well, all of us, all the researchers, in 70% of the genome, but there is still a 30% of the genome that it is not easy to analyze. Okay, so we need to take this into account. This is not a problem that has been solved yet, and there are, of course, lots of groups working on that aspect. This is the structure, the architecture of the data. So once we have our results from our analysis pipeline, we get some files, which are basically very complex text files, that then we index in some table format, and finally we index them in a database that uses big data technologies. Okay, the only message that you have to take here is that we are using big data technologies because the genome is very big, and if we would use just typical databases or Excel files or whatever, we wouldn't be able to integrate all this data and make it accessible and queryable in real time. So we need to use big data technologies such as the ones that have been developed by Facebook or Google, and this is how we can interact with data in real time. This is the graphical user interface to the RD Connect platform. So the way that this works is that first you need to select which patients, which individuals you want to analyze. So this is a window that can unfold. You can select the codes from the patients you want to analyze. You establish this relationship, which kind of variants you are looking for. You set some filters. And then when you click on the run query, you get some results at the bottom that identify which are the genetic variants, which are the mutations that are meeting the criteria that you specified. Okay, so if you're looking for a de novo variant, you're only going to be getting heterozygote variants. And if you know also that the parents are not affected, you will also look for variants that are heterozygote in the patient, in the index case, but in the parents are homozygous reference, for example. So you can set the filters in a way that it makes it easy for you to identify which are the causative variants among the 3 million variants. Each of us has 3 million variants that are different to the reference genome. And then for each of the variants, you get some information already in this table, but when you select each of these lines, you also get lots of information in different tabs in this top window. Besides this, also we have lots of links to external databases so that you can click and get more information on that, and then the doctor or the researcher can decide if this is really a variant that could be causing a disorder or not. In some cases, when it's a known variant or it's a variant on a known gene that could cause a certain disease, it's quite straightforward to identify that. But in many cases, especially for rare disorders or diseases that have not been studied a lot before, it is difficult to identify which is the causing gene, and you need to do a lot of studying and even do some experimental validation at the end. So what are the options that we have in the system? We have standard filters and annotations on the variant impact on protein, what is the frequency in control population. So we're usually looking for variants for mutations that are very low in the population in the frequency. Otherwise, many people will be affected of this disease and it will not be a rare disorder anymore. We have in silico predictors that tell us how damaging a certain change can be. And also we can have filters on gene names if we already know which genes could be causing that disorder, and also in genomic positions if we want to focus on a certain region because we have linkage analysis, for example, that have already positioned or identified in which region of the genome this could be located. We can also do filtering using a standardized predefined list of candidate genes of interest. We have options to upload and filter by customized gene lists. Some researchers already have a list of genes that they always want to look at first because they know that many of their patients have these genes affected. We have a real-time customized gene list generation using genes associated to disease through a meme or the symptoms. Okay, there are already genes that are linked to certain HPOs or to certain memes, so we can use this information to facilitate the work of the clinician or researcher. We can filter variants annotated already in ClinVar, a database of known variants causing disorder, and also by only pathogenic or likely pathogenic or variant of unknown significance. We can filter by regions with observed long runs of homozygosity, so regions that are homozygous in the genome, which are usually a result of consanguinity. We have also strategies to phenotype-driven prioritization of variants, because we know that certain genes are associated with certain phenotypes. If we have a good description of these phenotypes, we know which genes might be causing that disorder more likely. So we can use that information to prioritize our results. And then we also can have anonymized data sharing through matchmaker exchange and direct link to multiple external sources. I will talk a bit more about this in the next slides. This is the exomizer, and this is maybe one of the strong points of RD Connect, is that we are linking the genotypic information, the full genotype, so we can have the exome or the whole genome with the full phenotype or the deep phenotyping that the clinician has entered in a standardized format in the system. Okay, we have the HPOs, we have the LMs, we have the orders, and we have the genotypes. Once we do some filtering on these genotypes, we can use all this phenotypic information to try to prioritize, as I was saying before, for the genes that might be causing the disorder. Usually when you apply the first filters, you end up with 100 variants maybe. By using tools such as Exomizer, it can tell us which one of these 100 variants is the most likely to be causing that disorder. And then of course the clinician still needs to go and look into this information. It's not a clear-cut answer that that's the diagnosis. It needs to be worked a bit more. So until now, I've shown you that RD Connect enables full data sharing and analysis within the system for authorized users, and that's a question I was getting also in the chat before. Any researcher can register for RD Connect and can be a member. They need to accept a code of practice, and they need to sign an adherence agreement, which specifies what you can do and what you cannot do in the system, and what your liability by me if you do miswrong usage of that data. Okay, so this is all structure. You need to sign that before you can use the system. But if you access the system, then you have access to all the data in that system. There is an exception, which is that when you submit an exome or a genome to the platform, there is a 6-month embargo, meaning that the researcher have 6 months to look at the data first before anyone else does that, if they want to do so. This is to avoid skipping publications and so on. Okay, but afterwards, it should become available for anyone. So besides this data sharing fully in the system, we also have partial data sharing outside RD Connect in accordance with ethical and legal limitations. We have a couple ways of doing that. One of them is through the Beacon Project. The Beacon Project is an initiative from the Global Alliance for Genomics and Health. It is an international initiative. And the questions that you can answer with the Beacon are very, very simple. The question is, does any sample in your database have variant B? And then you answer yes or no. So you say, yes, at least one of the samples in my system has this variant, but you don't tell how many or who it is. But for very, very rare disorders, this might help to identify another source, another database, another system that might have another patient like yours, and that might be useful. There is a network of beacons that apply that, so you don't have to go and make the question to every system that applies the beacon specifically, but you can make a question to the network of beacons, and automatically this will ask all the systems that have applied this API, this interface, to answer the question and get the result on that question. Another way of sharing data beyond RD Connect is through Matchmaker Exchange. Matchmaker Exchange is an initiative from the ERDERC and the GA4GH again. And in this case, the question that you can ask to the participating nodes is if you have a patient with a similar phenotype and genotype as mine. Okay, so here is not only about the variant, it's also about the phenotypes. And what we have is that because, as Libby showed you, the HPO is like a tree structure and all the terms have a parent term, we can match patients even if they don't have exactly the same terms described. We have algorithms to do similarity indexes on human phenotype ontology and tell us that this patient is quite similar to this other one in terms of phenotype. And if these two are similar and the researcher has identified a likely causative gene in one patient and we identify another patient with the same likely causative gene and similar phenotypes, we can say that maybe this is really the causing one. We again are not giving out the name of the patient or even the code of the patient. What we do is put in contact the researchers between them so that they can expand this collaboration further. Then we have, and now I'm moving to the next part of the presentation, which is showing you how we have applied some of these into this call from BBMRI-LPC. BBMRI is the biobanking and biomolecular research infrastructure from Europe. And the project BBMRI-LPC is a project on large prospective cohorts. And within that project, we launched a call together with the Sanger to sequence 900 rare disease samples in collaboration with Biobank and RD Connect. The idea here was to provide free of charge whole exome sequencing and bioinformatics analysis to between 10-30 coordinated projects, each with 2-3 principal investigators from different EU countries. Okay, that was a transnational action, so it had to be from different countries, for a total of 900 samples. Okay, and the sequencing was to be performed at the SENAC and at the Wellcome Trust Sanger Institute. The idea was to promote the usage of biobanks for rare diseases, to promote the utilization of cutting-edge next-generation sequencing technology for the identification of novel causative variants and genes, to molecularly diagnose rare disease patients, and to promote data sharing for rare disease research to enable future discovery. So the key bits here is that the users or the researchers that were sending the proposal needed to have the informed consent from the donors. They needed to coordinate between different PIs from different countries, and they had to be willing to submit these samples into biobanks if they were not already there. At the end, we selected 17 proposals amounting almost 900 samples, and 10 projects were sequenced at the SENAC and 7 at the Sanger. These are the 17 projects that have been sequenced. You can see that they have a different quite different scope. Some are on Rett syndrome, other on gastric neuroendocrine tumors, albinism, we have eye disorders, gastroschisis, CMS, we have mitochondrial disorders, intellectual disabilities. So we have quite a broad area or field of disorders in that code. This is something that Libby already showed you before. It is I just want to highlight that we did go through all these will for this project, because as I was mentioning before, they had to have the consent from the donors, and they provided the samples to us to a place to be sequenced. They provided the sample then to a biobank, which is keeping record of that. This has been sequenced. They entered the phenotypic information, which we integrated with the genomic data. The raw data is going to go into the EGA, the European Genome Genome Archive. We haven't done this yet, but it will be done in the next step. And then all this together is available through the online analysis platform that I described a minute ago. And hopefully, in some cases, and we have done in some cases, we have already diagnosed the cause of some of these index cases. So I don't know why the background of my slides went crazy. I'm sorry for that. They should be all white, but for some reason it's all blue. This is the analysis status as a month ago, a bit less than a month ago, on the 10 projects that we sequenced at the SENAC. There are 7 still missing that are in the Sanger, and we are going to be analyzing them very soon also. This was a total amount of 548 samples from 237 families, and we had at this point analyzed already 219 of these samples, and we had identified 109 cases with good candidate variants, so 50% of these cases, and 79 of those were very strong. So we are quite confident for 79 of these cases, so 36% of those, that we have identified the causative variant. This is now, of course, on the hands of the researchers to look into this a bit further, do their own analysis also, and their own evaluation, and even experimental validation of these results. Some things I can tell you, again, unfortunate that all this background is a bit crazy, but all I can tell you from this number of samples is that when we have more HPOs, when we have a better description of the phenotypes, we do a better job in solving these cases. As you can see here, when we have between 0 and 2 human phenotype ontology terms, we are solving on only 25% of the cases. When we have between 3 and 5 human phenotype ontology terms, we are solving 38% of the cases. And when we have 6 or more phenotypic terms, we are solving almost 50% of these cases. Okay, so having more phenotypes is helping us to diagnose or to be able to reach easier to the molecular diagnosis of these individuals. Another aspect that I want to highlight is that in the system we have a way of estimating if there is consanguinity in the individual, so if there is consanguinity in the parents. So what we do is compute regions of homozygosity across the genome or the exome, and then we can look at how many regions we have of homozygosity in a given patient or in a given individual, and what is the total size of these regions. The higher you are in here, it is more likely there is consanguinity. And why is this important? It is important because in some cases we have a false assumption that we are looking for a de novo variant, for example, and in fact we are looking for an homozygous recessive variant because there is consanguinity. So this helps us to detect these cases when for some reason we might have missed some information, or we don't have it, or we have a wrong understanding of that, and then it can help us to correct the way we are analyzing the data. Or even the other way around. In some cases, some people think there is consanguinity, or they report there is consanguinity because there was consanguinity in their grandparents or whatever, but this is not the way to analyze the data at this point. And by looking at this and seeing there is no consanguinity in fact in the sample, then we change our approach and we can do a better job in diagnosing that patient. Another thing that we can do with the homozygosity settings is that we can filter by these regions. And when we are studying at patients that have consanguinity and that there is consanguinity in their parents, then we can use that to just look at these regions that are homozygous. So it is a way of filtering, a way of delimiting where we should find this causative variant in patients where there has been consanguinity. So you can see that from having 100 variants, if you apply this filter, you go down to only 9 variants. So you are making a big filtering to going down to only 10% of candidates that you need to look at, and it is very likely that you will find your answer among those. In terms of solved cases, we have solved cases of all kinds. We have some which are autosomal dominant, we have many autosomal recessive, but I also want to highlight that we have solved quite a few which are mitochondrial. This is important because we have used in this project, in some of the exomes that we sequenced, an exome capture kit that allows for also capturing the mitochondrial genome, which allows us to identify variants also in the mitochondrial genome, and some of the kits that you do exome sequencing with don't allow for that. So this has been a good approach. We think here it was the first time that we were doing it in a larger scale project, and has proven to be quite successful. I will not go into the type of mutations that we have identified, but as you can see, most of them are non-synonymous SNVs. And then just an example, these are known neuromuscular disorder genes that the group from Hans Lochmuller identified in his project in this school, and many of them were known, but he also identified some septins in one of their patients, which is a new kind of genes that they had never thought about likely been causing this kind of disorders, and now they have proved that it is indeed the cause of that disorder. So it is not only useful to identify causative variants in known genes, but also to identify new causative genes. And with this, I will leave it here with the big slides of thank you and acknowledgements, because this, of course, is the project of many people and contributions from people from many different countries.
Thank you very much, Sergi. So much information there. I hope people will be able to follow, and don't worry, you can, as I mentioned earlier, but for those who just joined us a bit later, the webinar is recorded and the link will be disseminated, so you can watch it again and also send it to your contact. So we had just a couple of questions coming in through the chat box, but remember you can also use your mic if you want, and I think Jean-Marc just sent you a link on how to use it as well. So Veronica is asking whether is fair data a requirement for the registries that want to join RD Connect? Maybe Libby or Sergi?
I can answer this one if you want. There is no requirement, but we are encouraging the registries and biobanks to become fair, and they are in fact bring your own data workshops that try to help in.
So you mean you are supporting them in making their data fair somehow, or is it training that you provide?
There is training provided mainly by Marco Ru's group and other groups in RD Connect. There is no budget to support all the registries and biobanks out there to become fair. So it is more about training than putting resources from RD Connect to make sure that every single registry and biobank can be made fair. Although there are plans also through Elixir, which is the European Bioinformatics Infrastructure, to be able to reach a broader audience and be able to
thank you. Where is the data stored physically?
So the genomic data and the phenotypic data, the rows of data, right? All this biobanking information and the registries, they are stored in their corresponding biobank and registry. We only have an aggregate of that information that is submitted by biobanks and registries. This is stored currently in GRAS in Austria. These aggregates, the ID card or the biobank and registry finder. The genomic data, we have two different places for storage. The raw data is stored at the EGA, the European Genome Archive, which is an initiative that has two nodes now, one at the EBI in Kingston next to Cambridge, and another one in Barcelona. And the processed genomic data, this is stored in Barcelona in the servers of the SENAC. So all this that I've showed you, the interface, this is stored physically in Barcelona, and also the phenotypic data through phenotypics is also stored in an instance in Barcelona.
So Deborah, I think you kind of answered that, but I don't know if you wanted to add anything. Ask where would she find the data protection recommendations. So you mentioned the code of practice of the code of conduct that researchers who want to use the platform and deposit their data need to follow. Is there anything else you wanted to add?
No, I think this should be available, Libby, in the main RD Connect page. If I'm not mistaken, you should be able to find this code of practice and adherence agreement. We are making it now much easier for users to register themselves in the system and to submit data. And in this framework we are preparing, we will have also easier access to these documents and easier means to register yourself and make sure that you are meeting all these requirements. Specifications. So I think for now you could find them in the main RD Connect page, but in the future it's going to be in the platform page with whole form to fill in.
Yeah,
know, maybe if you want
to confirm that it's all on the RD Connect website, we can maybe send the links to Virginie to send out.
No, I think that'd be informative. Yeah.
And also the data sharing charter that I mentioned before is also available on there, so we can share that with you.
Great, thank you. Nancy had asked who should we contact to learn more and get involved, and maybe also referring to, for example, patient organizations who have registries and who might want to join the RD Connect, so not ID card, what is it called now again? Sorry? How do you say?
The Registry and Biobank Finder.
Self-explanatory.
Is the new name. Yeah, but that's the aim. Well, to get involved, you can certainly contact myself or anybody at the coordination team for more platform-specific questions. Sergi is probably the better contact, but if you're wanting to, contribute information, then you can get in touch with us, and we can point you to the exact right person within.
Yes, indeed, there are many partners. So.
I also pasted now a link to, I hope it is still this one, from the Registry and Biobank Finder, in which you can propose registries of biobanks.
And I also just wanted to add that even though it's early development, but on the new and great RD Connect website, there's a section for patients and family, and we will progressively add materials that hopefully are specifically of interest for patients and family with anything to do with the different activities of RD Connect. So check from time to time when we add new materials on that as well, and there'll be videos and testimonies and webinars, hopefully. Veronica asked what will happen with RD Connect databases from next year when the project finishes, when the funding period finishes. Who will have the governance over them?
So maybe, yeah.
Okay, so
you go ahead.
Yeah, maybe you can give a general answer, and I can give you
specifics
about
so in general,
Go ahead.
We hope the project doesn't end, so just the current funding period will end. So we're obviously working hard to try and make sure that there will be ways to sustain everything. So we don't expect things to just drop offline or anything like that. And we're doing that for kind of every area of the project. So as Sergi mentioned, things are stored all over the place. So we're making sure that there will be ways for everything to continue. So hopefully in terms of the kind of user experience, there shouldn't be any change to that.
And regarding the governance, sorry, maybe, or is that for Sergi?
Yeah, so... I'm
not sure we finalised what the new governance structure of all the RD Connect sort of parts will be post the EU funding, but certainly I think there's a willingness from all of the partners to remain involved and to keep everything going. So hopefully it will be quite similar to it is now. So at the moment we have a management committee made up of work package leaders, and we have the advice from the PAC, and hopefully we can try and sustain these in one form or another, but we don't have all of the kind of...
And if I can add just on to that as well, we have discussed obviously the governance of the and maintaining the kind of patient expertise that they've been involved in the project, and to have still some of that remaining in the future when the funding period ends, and when the project takes a different form, but will still be there. Sergi?
Yeah, I just wanted to say that we are I'm confident that the data will not disappear after the funding period of the end of 2018. We already have secured funding for all the genome phenome data from different sources that will ensure that this is going to go on at least for 5 more years after that. I mean, one thing is maintaining what we have, the other thing is how fast we can go into developing new things into the system, right? So one thing is maintaining, the other thing is developing new things, but we are quite confident that this is going to go well. It's going to be sustained. We are looking at different scenarios, and we are trying to find even other ways of making this sustainable, even for the longer term. What I can say is that there are some big projects that have already committed to submitting lots of data into RD Connect, and with this also comes a bit of funding to make this possible, which means that also this helps to promote the project further.
Thank you. So almost all bases covered. Thinking for the future. So Veronica is happy with the answer. She said thank you. Does anyone want to maybe speak and ask a question through the mic or through the chat box? I think we went through all the ones that were written so far. Maybe not. I don't know if Libby or Sergi, did you want to add anything that you thought of maybe afterwards, or?
Well, I think that the point that was raised before about RD Connect being usable or being able to be used by patients, that's a very good point. And you and me, Virginie, have discussed about this previously, and I think that you have a good idea of what the patients need in that sense, or what they would like to see. So I think that's something that we should try to work on at some point. I don't know exactly under which umbrella or with which framework, but if there is an interest from the patient associations to make this a reality, we should try to push for this, and I think EURORDIS
No, it is a good point, and it's something that we can also discuss with the members of the patient advisory council already. Maybe as a group, next time we do meet, we try to meet every few months, and maybe take it even wider to find out what patient wants or needs as well. So it'll be good to get their input and to see the kind of level of need and demands. Yeah. Mm. Hello. Yes, I can hear you.
This is Nanda from Sweden. I have a question.
Yeah, if you can speak a tiny bit louder, maybe.
Okay, I can try. How about now?
Yeah. Yeah.
Yes. How many doctors around the hospitals in Europe are aware of this project?
So did you hear, Sergi, how many doctors are aware?
Of the project?
Yes. Yes, I don't know how many are aware of it. I know that we are working a lot now with the ERNs, with European reference networks. The ERNs are the expert networks that integrate a lot of the expertise around different groups of rare disorders. I think there are 23 or 24. I think it was 23 before now, and now it's 24 ERNs. And we are working on them through several channels to make sure that the project is known, and that we make it also, that we customize it somehow to make sure that it's useful for them. And we are working on
it. Yeah, and that's a big outreach really, because I think there's at least, if not more than 900 different healthcare professionals and centers involved in all these different ERNs. So it means it's reaching more and more clinicians and researchers as well. So that's definitely a step forward in making RDConnect known, and the more it be known, the more it be used. Does that answer your question?
Yes. Absolutely.
Great.
I speak to doctors every week, and I wonder if I should tell them about the project, because we meet people with rare diseases in different ways.
Yeah. That would be great. Yes, definitely. I think it cannot hurt. The contrary, if they don't know about RDConnect, they might be able and be interested to participate.
Yes, that would be great, because it's a great platform, and you can participate by webinar, because many of the doctors say we can't participate, we can't travel around in the way we need. So webinar is a great way to participate.
Good. I will circulate the link of the webinar, and you've got also Sergi and Libby's contacts if you need more information. So don't hesitate to contact any of us on it. Thank you. Anyone else would like to ask a question? I don't think so. I can't hear anything.
Veronica is typing.
Ah, someone is typing. Let's just wait a couple of minutes. Is any of this data used for genetic research as well, or just for clinical use? So is it just to provide a diagnosis or develop a treatment, or can it be reused for research?
It's obviously, yeah, I mean, in fact, RDConnect is basically a research platform. So it can be used for diagnosis, but it's clinical it's clinical research, really what we are doing here. So we are using that to discover new genes and new mutations into the system, which is research, and many of them need to be validated experimentally afterwards. So yes, it's used by clinical research, and hopefully some of these new variants also can be used then to identify likely new
and I think, when we talk about diagnosis, the line is definitely very blurred between healthcare, a clinic, the clinic and research, because they all kind of feed into each other. So it's difficult to make the distinction. It's for both, but I don't know if that answered your question. I hope so. Veronica is typing. Are you considering partnering with other entities in order to search for treatments or to develop treatments, I guess? Is there more partners being invited to join, or do you collaborate?
So that's not something we have officially done yet, but that's something we have discussed in some of the meetings, EMC or some of the annual meetings, how we can make use of this information so that it also helps identify new treatments or repurposed drugs. That's definitely under the radar, but it's not something we have actively acted on, although we do have some people interested in knowing some of the specifics to be able to see if there's something they could do with that. But yeah, it needs to be studied a bit more. Also, to what extent we can share this data with even, for example, if you would think about a company, so far no companies can access this data, but would it be possible that you share some information, provided you don't share any personal information? You just say, this is the variant, this doesn't identify anyone, but can you design a drug for this variant or for this gene? That's something that we could potentially do, but we have never done, and this also would have to go through the committees and the ethical and legal work package and so on. But
okay, that's a very good point. Okay, she's typing another question. Thank you.
Welcome.
Okay, if there are no other questions, maybe we can close the webinar here.